So with the permission of chair and co-chair, we'll now move towards our scientific sessions. Our first speaker of the session is Dr. Professor Arif Shaikh, who has come all the way from America. He is clinical professor, medical director, and chief nuclear medicine at Kettering Hospital. He is director of targeted radionuclide therapy. He has also led multidisciplinary thyroid and endocrine tumor board and developed targeted radionuclide therapy clinic. He has extensive teaching experience of trainees in major specialties, including nuclear medicine, radiology, cardiology, radiation oncology, and general internal medicine. I request Professor Dr. Arif Shaikh to please come on the stage and present with his talk.
Thank you very much, ladies and gentlemen. Good afternoon. Assalamu alaikum. It's always an honor to be invited and always a pleasure for me to come over and present. And a special honor that I am the first speaker kicking off the conference with a plenary session based on trailblazing trends in nuclear medicine. I will say that we are moving at a very breakneck speed as to what's happening. If someone tells me they're keeping up with the literature, they're lying. I do not believe them. The literature is moving extremely fast. If they're either not telling the truth or they are not working. They're just spending their time reading and not doing any practical stuff because things are happening very fast. I have no disclosures for this event. So where is it happening? It's happening everywhere, in all aspects. Oncology, cardiology, neurology. It's happening in the clinical sciences. It's happening in the basic sciences. Radiochemistry is developing. Radiopharmacy is developing. This has all happened within the last 6 years, and the last 4 years, and the last 2 years. It's accelerating at an enormous pace. Technological developments. There's a paradigm shift in the types of services we're providing. There's the accelerated knowledge base that we're accumulating. And what's driving this is, let's face it, is big pharmaceutical companies pouring in cash and is driving the whole field forward. So again, some of the memos from within the Society of Nuclear Medicine, we can see that just in the US, the trends and projected trends in the market are big. There's nothing like this going on in medicine anywhere or has been seen for a while. Now, part of that is we're a very small field and therefore growing very rapidly. But if you just look at this is all the products that are being developed. As you can see, there are basically 4 circles. These are the therapeutic products. These are not necessarily just the diagnostic products. So the outer circles are in phase 1, the inner circle phase 4. The only 2 that have been FDA approved in the US are Lutathera and Pluvicto. All those other products have popped up in the last 2 to 3, maybe 4 years. Okay? Now I will tell you this. About 10 years ago, I visited the National Institute of Health, the NIH, in the US. And there was a conversation going. Make a long story short, they told me, we have 3,000 ligands waiting for somebody to touch them. That was 10 years ago. This is nothing new. Because there's pharmaceuticals driving this, everyone's jumping to the old stuff and maybe modifying it and stuff. So if you're thinking that number is big, wait till the rest of the 3,000 show up. So yeah, we already know there's water behind the dam. So what is the latest new trends? Why I say the latest new trends? Because there are new trends all the time, but that new trend is changing constantly. So these perhaps latest new trends, again, there's nothing really mysterious about them. It's multimodality imaging, molecular imaging, technological breakthrough. In therapeutics, we're already thinking about multi-radionuclide therapies, multi-targeted approaches. Dosimetry is becoming very big. I've given talks in dosimetry in the past. People would kind of look at me and say, okay, very interesting, but all of a sudden, It's something that's come into our faces. In the U.S., these are the radiopharmaceuticals that are already approved for oncology, cardiac, neurology, and the ones in development. And again, you can see based on that, these are already this list is obsolete, right? Because it's growing so fast, I can't keep up with it. So just case in point, estrogen imaging for breast cancer. It's Breast Cancer Awareness Month. I thought it would be appropriate to just show a slide. Patient had an FDG staging PET scan done, which is the bottom row. You don't see much anything. And then it had an estrogen PET scan for a lobular cancer. And you can see their foci of uptake in nodules that look very, very nonspecific on the FDG images. The images are required just 3 months later, and they're clearly there's metastatic disease. So completely change the staging of this particular patient. Last year I had a presentation on methionine imaging and amino acid imaging. And again, here's a patient who had FDG, the amino acids at the bottom, the FDGs at the top. We can see the tumor has different expression of metabolic tracers. And yet, by the same token, another patient may have methionine and FDG uptake. These different expressions mean different things. And we know that to some extent, but what they mean for each type of tumors and when and why is becoming much more relevant. This is a patient who had an astrocytoma and had a recurrence of an astrocytoma that was picked up on the left scan on a PSMA PET scan, okay? So we have to be aware of these things as we're reading these scans. That was resected. It was evaluated with a methionine image in the middle. And you might be able to tell that there's a little bit of a dark sort of foci at the edge of a resection cavity on the methionine images, which are not there on the FDG images, which are sites of recurrence. So again, we have to understand what all the receptors are and which tracers are going to show up in what pathologies. PSMA is just the opening shot because everybody's interested in FAPI, right? And FAPI, as far as we know, is 28 tumors and counting and has great expression, and yet it is also somewhat discordant with FDG. So how many other tracers is going to map with, not map with? So we really have to start thinking about the various receptors we're imaging, not just the pathologies anymore. I think we're all moving away from the paradigm of a histologic pathology and more a receptor-based pathology as we look at things. So here's a list of theranostic agents out there, at least in the US, that have been FDA approved versus those in development. And I mentioned that list is already obsolete because that list is growing at a breakneck speed. So what are we interested in? Yes, we're starting to administer it, but everyone's getting interested in this concept of dosimetry because data is coming out showing, well, if you give higher doses to certain patients, you may get comparable toxicities, but you get improved outcomes. So in this case, for example, the patients getting higher doses of PSMA did better in overall survival than lower doses. This is just, again, dosimetry in another context, as people getting liver-directed yttrium-90 microsphere therapies, again showing that tumors respond better to when there is more radiation given to them. And again, these concepts aren't new. I mentioned I've presented on dosimetry in the past. We've done that work. I did that work when I was in training 25 odd years ago. This is I-124, we did dosimetry of the or thyroid cancer patients. We did dosimetry of normal organs, trying to see what kind of doses are actually being delivered. So we have the techniques to measure the doses, but is that all? Well, there's a couple of some literature out there. I had an abstract which was presented on the left side, and another paper who independently published their data on the right side. Both showed that whereas there was a lot of variation in the amount of dose given and If you compare gigabecquerels versus gray, there's still a lot of dispersion, but there seems to be a trend on a univariate analysis where gray is a better predictor of toxicities than gigabecquerel. That would make sense, right? It would make total sense. But we've gone and looked back at data. This data is unpublished, I'm going to show you. And we did a multivariate analysis. We looked at gigabecquerels, meters squared, creatinine value, age, sex, that kind of stuff. Came and looked at a simple linear model. On a simple linear model, actually, the gigabecquerels do better than the gray in terms of predicting toxicities. And this is the thyroid cancer population. So what does that mean? That means our models aren't quite right. You know, there's still some more understanding that we have to do. Everything I'm presenting could be a talk by itself, so I don't want to get into the nitty-gritty details of why this may be the case. But we know this is the case because even the models of the physics models that we're putting together and various experts have put together show that you can take the same data and come out with different predictions, whether it's an absorbed dose model or biologically effective model, equidose 2, that kind of stuff. So we still have to pin down what are the physics of this. We still don't have a good, solid understanding about that. Okay, so stepping away from that for a little bit, what about the targeting of the tumor itself? Well, right now everything is good. We're just giving PRRT. We give a little bit of amino acids as a protector of the kidney, but we're not targeting the tumor any differently. We've known about multi-step targeting for well over 2 decades now, where we may give an antibody attached to something like streptavidin. We give a clearing agent to clear any remaining antibody in the blood so that just leaves the targeted antibody, and eventually give biotin, which has great avidity for streptavidin, and the biotin has a radioisotope attached to it. So it will very specifically target the tumor. So it's a multi-step process in how we're targeting the tumor. But again, radiochemistry has developed, and there are neurolinkers and agents. SADA is one of them. Self-assembling and disassembling domain. This is not by any means the only one. There are several of them. I'm just showing you examples where this provides certain advantages to just the multi-step targeting that I showed. For example, this is just showing you a graphic of what it would look like, because your toxicities are going to be based on your tumor to blood ratios. So if you were just to give an antibody attached to a radioisotope and it floated around and got to the eventual target site, you can see on the left side the the tumor to blood ratio is relatively high. But if you start getting into these ideas of multi-step targeting and clearance, you can see that improves. Eventually you get to this sort of the SADA type thing. And again, these are sort of theoretical models that you will improve your targeting much better and reduce your toxicities. Not only that, these are antibodies. So, and we have targeted proteins. We have different type of proteins we're putting together, different domains we can modify. So just that is changing, and how we use that in our targeting and our radio labeling and what we use it for is going to be very important. And as these proteins or antibodies become more sophisticated, remember, they're going to have activity by themselves, irrespective of the fact that we've also given a radionuclide on top. So, so we give these treatments. We're doing multifractionated treatments. We actually did, again, as I was a resident, I participated in clinical trials for, which is the C2 Chimera G250, which is now called girentuximab. It's coming through for renal cell carcinoma. These are 3 different courses for a patient. So a patient would come in, get a course of fractionated treatment, come in a few months later, get another course of fractionated treatments, and before each course, they would have a dosimetry, just a single dosimetry where we just look at one clearance, and based on that, try to predict what the fractionated approach would be. But again, since this was an antibody, you can see after fractionation that at some point, especially as you go through the third course, the dotted lines show you how much the actual dose is straying from the predicted dose, which is the solid line. So again, we are not quite able to do dosimetry, or we are kind of getting into the ballpark, but really understanding how to get those exact numbers is even a bigger challenge than just the physics. So with that being said, what are we looking at? We're looking at different disease states. We're understanding that we're starting to get all these biomarkers, where to use the imaging, when to use it, and even within the tumors, expressing different gene types and receptors that we're going to be using different types of therapies to treat these patients, and even imaging for that matter. And even within a tumor type, are we looking at a mild, an aggressive form, or a highly aggressive form? Should we be, FDG is probably not going to go away. I mean, FAPI is coming up, but we have an inherent understanding of FDG. FDG is probably going to be our base understanding of tumors as much as thyroid cancer is our base understanding of theranostics. So that's probably not going to go away, that understanding. It's just that things that we've never thought about before, even in thyroid cancer, we should start thinking. I take some of what I learned from prostate cancer back to my thyroid cancer patients, which is something that I feel that our endocrinology colleagues do not have that advantage of being able to do so. But throwing on top of all that, we know that there are biomarkers, there are blood markers that are being developed that show high predictivity in being able to predict whether a patient will respond to PRRT or whatever other therapies that they are. So throwing this into the mix with dosimetry and all the other clinical markers may help us predict. And that's what goes back to that slide I showed you, that if you have a multivariate analysis, you might be able to better predict how the patient's going to outcome, and it's not just about just the physics anymore. I was involved in the phase 1 clinical trial a little over 10 years ago, because where we did this is lutetium J591, which is an antibody for prostate cancer. This was a phase 1 analysis where a trial where we did lutetium J591 with docetaxel. So it was done fairly concurrently. And this is probably going to be the wave of the future, that we're probably going to start combining therapies as we go along. J591, by the way, is now also making its way through phase 3 trials. It's going to be an antibody. Pluvicto is given 6 treatments ideally, but J591 may have a little bit of a different profile yet to be seen. And we're starting to combine them with other traditional therapies, such as radium, giving radium to some of these patients along with lutetium, not concurrently, maybe sequentially. I've done that with a couple of patients. I've been impressed. It's interesting. All of a sudden, I feel I have tools to offer the clinicians where everything else seems to have failed. The patient thing seems to have worked. So again, we're looking at the safety and efficacy of this stuff. So not are we only looking at that, we're looking at a combination of therapies. We have to understand what the medical oncologists are doing, what their data shows, and how our data compares, and how are we going to combine our thoughts and modalities in treating the patients. So again, the future is wide open in regards to that. Will dosimetry be sufficient? As I said, we probably need more data just than dose delivery. And how exact do we need to get? Is it that important to get an exact number? And is it practical to get that exact number? That's an issue. Well, because, the whole thing is that the radiation that you're giving, you may not need to give the killing dose. You may just need to give it enough so that it is perturbed and that it responds to chemotherapy or immunotherapy better. You may start causing abscopal effects, as a result of that. So it may be a combination approach. We may not need to know the dosimetry exactly. Just kind of get an idea of where it's going. Maybe get to understand toxicity better if we get the better models to predict that. And that's just oncology. What about neurology and cardiology? Big developments going forward. As quantification is picking up in cardiology, radiopharmaceuticals are coming, neuropathologies that we're starting to get into. Start thinking about things that we never thought of that we could before. This is an interesting, an article that recently came out. It's just sort of an illustration of the history of nuclear cardiology, all the way from blood pool imaging, coming down to technetium SPECT and PET imaging, and now getting into viability, sarcoid, and so on and so forth. So nuclear cardiology is certainly in development, looking at a lot of different disease states, a lot of interest in developing various types of radiopharmaceuticals. The big interest, and now very clinically viable, is looking at corneal flow reserve. I will be presenting some cases later in the conference this year on how to use corneal flow reserve. The one big thing is, the dreaded triple vessel balanced ischemia, where the scan looks fine, but it turns out they have balanced disease. That's where it is. But once you start using this tool, you realize it is much, much, much more than that, and in the management of the patient. And I hope to be able to show you some cases for that. Amyloid imaging in cardiac, it's felt it is an unrecognized disease. We're screening a lot more patients. We're doing it on SPECT side. There's a huge movement to do it on the PET side. So that's in the making. And then there's neurocardiac imaging. We could look at cardiac imaging to help define certain type of neuropathologies. So we're looking at different pathologies at different places to be able to make diagnoses. And speaking of neurology, the big thing now is Alzheimer's disease. There are now treatments available to treat Alzheimer's. I think these are just the beginning drugs. They will probably get more sophisticated as time comes along. But we're doing FDG imaging, amyloid imaging, tau imaging is becoming important, and knowing what those pathologies mean in context, knowing the entire neurology of that itself is a field of discovery. And there are neurotracers, again, that look at both tau and amyloid together. And now we can look at not only dementias, but things like chronic traumatic encephalopathies, something else I'll be presenting on a little bit later this conference. And then things get even more interesting. If you look at the bench literature research, there's already therapeutic concepts of targeting beta amyloid plaques for Alzheimer's disease with radionuclides and breaking them up with that. So this is going to be very interesting if it develops. Personally, I want to definitely see more data, because part of the issue is if you unleash alphas all over the brain, you have neuroinflammation. That's not a good thing. But let's see where this goes. Or the treatment of infectious diseases. We're starting to develop superbugs to our antibiotics. We're losing that war. We're going to lose that war if we keep going to where we are. And this may be a way to at least bypass some of that. So again, theranostics in the area of antibiotics and infectious diseases could be an area that we would see development further on. But all this requires scanners and equipment to be present all over the world. And SNMI in conjunction with all these societies, we're starting to keep a track of where the scanners are, what's going on, who's developing, and trying to see, kind of predict the needs that are going to be needed in order to handle the world's population with all these diseases. And just the technology itself is developing. It's not just the presence of scanners. Now we have full-body PET CTs, we have digital SPECT CTs, PET MRs, so on and so forth. Okay, so, and these newer technologies are already showing us that we can start doing things with this stuff that, again, we had not thought about doing before, or we can do better and can make it part of our clinical practice. So, whereas a full-body PET scan at this point is considered very academic, it's quite possible in the next 10, 15 years it's going to be adopted, or maybe even earlier than that, by less academic environments. Because not only are we going to have PET scanners, but we're beginning to look at things like something called prompt gammas that can happen with PET, and look at not positron imaging, but positronium imaging. So there's already work in Poland with regards to that, and this is in terms of looking at glioblastomas. So this should, so again, more technology and more understanding of what a positronium is. I won't go into what a positronium is right now. Again, we can go into that a lot more at some other time. So what are we looking at? Here's not one patient, but these are different patients. But these are all different agents that can look at prostate cancer. Okay, so here I'm just showing you 6 different tracers that could look at prostate cancer from different aspects. Some of them can be used therapeutically, some of them are just purely diagnostic. And each of us tells us something a little bit different. So picking and choosing, so now the clinician will come to us and say, what do I choose? Which scan should I go for, right? You can't go with all 6. Because these are the trends, this is a practical issue. These are the trends in costs of diagnostic testing. You can see imaging way outstrips everybody else. So we can't just keep doing more scans. We have to get smarter about it. But guess what? We're also getting smarter on the technological side. Because again, on the basic science side, we've seen proof of concept that again, using prompt gammas, we can image 2 different PET radioisotopes at the same time as simultaneous imaging. So that should be very interesting because that would mean we don't have to do, say, separate FDG and dotatate scans. We could probably just give it together and get 1 scan out of it, or 2 or maybe 3. Okay, that was really not considered feasible several years ago, but now at least on the basic science side, it seems like there may be some development in regards to this. Some newer radioisotopes to watch, and again, with all this development, we want to be able to show used newer radioisotopes. This list is what? It's obsolete. Because now you look at the literature, you'll see all sorts of radioisotopes sort of just percolating through. These are the ones that are sort of making their way through, that you'll see that are already in the clinical trials phases and are being talked about. But there's many other radioisotopes that are coming through. And these are the current facilities in the world that are the major reactors that are producing the radioisotopes that we're all using. We're relying on them to provide us our raw material at this time. There are other facilities, of course, as well, but these are the major ones. And because of that, there's this whole reactor generator supply model. It's global. It's susceptible to global disruptions. This day and age, you've got to think about the political issues, economic issues that are taking place, social environments, all things that can disrupt this at any time. So we are looking at, we have to develop reactors. And again, the SNMI and everyone else are keeping track of this. How many reactors are there per continent? Where are they needed? How many cyclotrons are present? Where are they needed? What are they going to be able to supply? So the continuing challenges are, we're in the midst of a technetium shortage, at least global. When I left home, that's what they said, and what I fortunately got an email saying, well, we might just skirt it a bit because we happen to have gotten a generator that was a little bit more generous than we had anticipated. So we might write it out a bit, but it's there. But I used to remember technetium shortages way back when I was training 25 years ago. So this is something that we have to overcome in order to provide the patient services, the consistency in patient services. But again, the issue is we can produce lead, but does lead have to go to everybody? Are we going, if there's a lead-based isotope and an actinium-based isotope, maybe we might be able to do a very similar job. So we may not have to ship lead across the world just because a certain vector carries lead, and that vector could be carrying actinium. But I do know this, that again, what's going to drive a lot of this is actually private market entrepreneurial forces. There's a small company called Ionetics that had made a small ammonia cyclotron that have jumped in and are privately starting to produce actinium for clinical trials and things like that. And there are a lot of these other little companies that are jumping in with reactors and cyclotrons to be able to make all the different isotopes that are being asked for. So what about global distribution? Again, is this going to become a regular service like in all hospitals, like, you know, most major hospitals or even moderate-sized hospitals have a chemotherapy unit? So should we be having a theranostic unit everywhere? You know, is it going to be in their rural hospitals? Something to think about. We are seeing in the US, certainly, and other places around the world, the rise of private practices offering theranostic services. And is this going to widen the economic divide between the advantaged and the disadvantaged countries? And even within a country, is it going to split social levels as a result of that? Again, another map. This is now Lancet has, in oncology, put a task force together to look at this issue of radiopharmaceuticals. It was just published in September, so this is really a new data. And they're looking at this aspect. For example, this is looking at PRRT and MIBG treatment for neuroendocrine tumors. And again, trying to get a handle of where things are and how things will need to develop over time. So these are all challenges that we're facing. Everyone knows this tide is coming, but the task is daunting by the same token. And healthcare disparities, as I just mentioned. The picture I showed you, a lot of folks know, people know, this is reality for a lot of people. This is dental care for this patient. We are an expensive modality. We are an expensive service. You know, so how do we balance priorities even within the U.S.? Even within the U.S., how do we balance those priorities? So, again, one of the other things. This is Dr. Sartor. I happen to have met him a long time, but he's one of the PSNM people. Basically, he says that, you know, PET scan might be useful because we can use it to target what we see. He's made this statement relatively recently. We've known this for several decades. We inherently know that that's what we're offering. This is something our colleagues are just beginning to understand. There's a rapid rise of radiation oncology getting involved in this. Nuclear oncologists are slow to get up just because we're a smaller group. But my take on this is very important, you know, especially if radiation oncology says, oh, this is all radiation-based. Theranostics is actually not a radiation-based therapy. It's rather an immunotherapy with a radiation component to it, okay? That's important. So, you know, I don't, when the radiation oncologist says, oh, it's a radiation-based, no, it's not. I want to work with my medical oncologist because looking at multi-step targeting and everything like that, antibodies, that's the main component. I'm adding the radiation component to their therapies, and then we shall see where it goes. Yeah, we may have to share facilities. And this is, again, is a snapshot at a talk, radiation oncology. They're looking at this very closely. Oh, this is big money for us. So we have to establish the narrative here and understanding what this is. Because we know what's coming. They don't understand. They see what's now. They don't understand what's coming. We know what's coming because I did it 25 years ago. We did this 25, 30 years ago. We know all that stuff. So my parting thoughts is, you know, it's important because there's so much money coming in. Radiation oncology is big. All these other places are big. They may just come in and take us over and drive us out of the field. We have to prevent that because we already have an inherent understanding of what this is. Are we going to subspecialize? In 5 to 10 years, I may not read neuroscans anymore because I may have to concentrate on oncology. Like I said, we have to establish and control the narrative. Let's think about it. We're going to split, you know, people are going to urinate radionuclides into the environment that we give them. There's an environmental impact, okay? People are going to start asking questions. Then the conspiracy theories are going to start, right? So, again, be prepared. How do we diffuse nuclear technology in today's global political environment? You know, there's a lot of tensions in the globe as a result of that. there are healthcare disparities, ethno-racial, economic injustice, things like that. And I hope that kind of gives you an overview. Again, a lot of thoughts, a lot of things to go into in depth, and I hope the rest of the conference will answer those, some of those questions for you, or perhaps bring up new ones. But most importantly, I hope there was food for thought. And with that, I thank you very much for your time.
Thank you, sir.